Okay, without any further ado, I welcome you to the second half of Biology 107 with Dr. Cindy Malone. She said she's mean. Don't believe her.
I'm not fun. Dr. Steino's way more fun.
No, I'm not.
Sorry. No. Oh, she's going to spy on me. She doesn't trust me. She doesn't know what I'm going to tell you guys. No, I'm not. I'm plugged in. Oh, wait a minute. Doesn't work. Much better. Mood lighting, dude. I look so much better in lecture capture in this light. I asked for the fuzzy filter, but they wouldn't do it for me, so. Okay, so I have no idea what goes on in the 107 lab because it's not coordinated with the lecture. Have you guys done mitosis, meiosis with them yet? Okay, then that's better. Okay. So I hope the quiz wasn't too hard. It was really just supposed to get you in the mode, and Stein had it posted, so I just put questions in it. Okay, so because, right, with the quizzes we get to drop 4 at the end, so if we didn't have one today, then you'd only get to drop 3. So I figured we want to have a quiz all the time. Okay. So we're going to talk about mitosis and the cell cycle. Okay, we're going to sort of start from scratch. I hope you guys find some of this somewhat familiar from whatever place you've been before this, right? And both of the cell cycle is essentially cell division. Okay? Most cells in our bodies as adults are not dividing. They're just sitting around doing their job, okay? The only cells that are really continually dividing in our bodies are blood cells, right, from our bone marrow. So our adult stem cells are constantly making red blood cells and white blood cells and platelets, all the things we need for our immune system, carry oxygen, all of that kind of stuff. So those are constantly doing cell division, right? Some of our epithelial layers are doing cell division. Replace the lining of our stomach, the lining of your intestines, right? Your skin, right? The few layers on the outside are dead, and those go away, right? We keep regenerating that. And our hair follicle cells, the stem cells in our hair follicles, are constantly dividing. Everything else is just sitting there doing essentially nothing except doing their job. Okay? A few cells here and there in your organs regenerating. Most of those are adult stem cells again, right? Replacing damaged cells or lost cells. Okay? And so all of those tissues are doing mitosis. So mitosis is cell division for normal, everyday somatic cells. All of our body cells, with the exception of what type? What type does meiosis? Yes, the sex cells. Those are the only cells that go through meiosis. Okay, everything else is mitosis. So the only time you'll ever answer meiosis on a quiz or a test is if we're talking about making gametes, right? Making sperm, making eggs, right? And for females, our eggs are all essentially made and stalled during meiosis when we're in utero. Okay, so ours aren't doing any division. They're just maturing. And if fertilized, they go through the rest of meiosis. Whereas boys, on the other hand, are kind of constantly doing meiosis. Right? Fresh batch all the time. Right? Okay. So generally, for almost everything, we're always talking about mitosis. Right? Once we have an egg and a sperm, and those guys come together, look at that. Ooh, help. Sorry. Right? Then we're back to mitosis, mitosis, mitosis. So that is confusing to some people. They keep wanting to say meiosis at that point. Meiosis gets us to the egg and the sperm. Once those two come together, it's We have one single cell, and then we do mitosis a gajillion billion times to become the adult organism. At that point, the only meiosis going on is if it's a female mammal making the eggs while it's doing development, blah, blah, blah. Okay? Everything else is mitosis. And so those are the two main types of cell division. Mitosis for everything except making gametes is meiosis. And for most organisms on this planet that are diploid, all the mammals and vertebrates, things that we think of as animals, we have two copies of every chromosome. Okay, so human beings have 46 total chromosomes, right? We have two copies of number 1, two copies of number 2, number 3, blah, blah, blah. Boys have an X and a Y. The girls have two X's. We split those in half. One number 1 goes one way, one number 1 the other. So each egg has 23 chromosomes, one number 1, one number 2, one number 3, one number 4, blah, 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 and an X. Sperm, one number 1, one number 2, one number 3, blah, 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 an X or a Y. Okay? We don't want two number 1s in one sperm and two number 2s over here. So it's not just a random split of the 46. Oh, we just need 23 over here and 23 over there. Right? You need 1, 1, 2, 2, all that way down. Because then the beauty of that is upon fertilization, we're back to 46. Two number 2s, two number 3s, two number 4s, two number 5s, blah, 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 blah. An X and an X if it's female, an X and a Y if it's male. And then we're just mitosis up the wazoo. Okay? Okay. The wazoo is the bunger, the bunghole. Yes. The hoo-ha is the JJ. Yeah. Okay. Just scientifically, that's scientific terms for those. We'll continue to use scientific terms for the rest of the semester. Okay. So blah, 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 the real definition, right? Division of genetic material. What's a genetic material? DNA, right? The chromosomes. In our nucleus, right, genetically identical to the parent cell. So in this case, we have 46 chromosomes in humans. We want each of the two daughter cells to have 46 chromosomes, right? We don't want it cut in half in a normal liver cell. Otherwise, right, every time you need a new liver cell, each one then has 23, and then if that divided, it would have 11 and a half, and then pretty soon you have none, right? So asexual reproduction technically applies to mitosis, right? Anything called sexual means a fusion of egg and sperm. Anytime you are doing cell division or making another copy of any kind of cell, we would call that asexual reproduction. And again, the real term for the body cells, everything but the gametes, is somatic cells. And so if you see that term, those are just your normal skin cells, liver cells, brain cells, rectal epithelium. Damn near killed them. Okay. And then the real definition of meiosis that our book says, right, division of genetic material from the nucleus. We want daughter cells with half the amount of DNA. Production of gametes, the sex cells, the eggs, the sperm, the ova. I don't know, what else do they call it? Okay. What? Swimmers, yeah. Okay. So for mitosis, let's see what's next, since I don't remember. Oh, okay. So for mitosis, if we want two daughter cells with the same number of chromosomes, what do we have to do before we do the separating of putting chromosomes to either side to make two new cells? Replication. We have to copy the chromosomes. Right? Somehow we have to have more than 46 in order to split in half to get each to have 46 full chromosomes. So for mitosis, we have to copy the chromosomes. if you think about meiosis, you shouldn't have to copy the chromosomes in order to just take 1 and 1, 2, right? You're just splitting them in half. But as it turns out, you do have to replicate before meiosis. It seems dumb, it seems like a waste of time, and maybe it is, but if you think evolutionarily, all organisms began as asexual, just doing mitosis to make more of themselves. You're essentially cloning yourself, just like bacteria do still today, right? They have one circular chromosome, they double it, one to one side, one to the other, down the middle. Woohoo! Now there's 2 of me and we're exactly the same. Okay? So the process of complicated cells with chromosomes to get through mitosis that requires replication, right? That's how it all worked at first. And so when evolution happened to make sexual reproduction, the formation of those cells still followed that same pathway, replicating the chromosomes, going through that process, but in the end you end up having it, which we'll talk about that process. So what I'm saying is that the beginning steps of mitosis and meiosis are exactly the same. And they call that first phase interphase for both. And it's the same for mitosis and meiosis. Okay? 800 years ago, when I was in college, they didn't separate interphase into its own entity. Right? They called it mitosis, and it was interphase, metaphase, or interphase, prophase, metaphase, anaphase, telophase, cytokinesis. That was mitosis. Meiosis was interphase, prophase 1, metaphase, blah, blah, blah. Now they've made it easier for you guys and pulled out interphase because it's exactly the same. They used to think it was different before mitosis and meiosis. It's the exact same process. So if I say mitosis, that really just refers to the separation of the chromosomes. Prophase, prometaphase, metaphase, anaphase, telophase. That's mitosis. Cell division has to start with interphase, then prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis as a separate entity. So they've changed sort of how we categorize those. It's the same process as it was 800 years ago. It's just they've pulled the categories differently. So if something's referred to as mitosis technically by our book or by me, it's really just those phases, the actual separation of the chromosomes. The copying occurs in interphase. We'll talk about that too. Right? So the copying of the chromosomes is part of interphase. It's not part of mitosis, not part of meiosis. It occurs in interphase. Right? And then mitosis or meiosis, whichever we're talking about, then the cytokinesis. Right? And so then that's a lovely little picture. Ooh, we started 1. We make 2. When we talk about chromosomes, me, I think all of us, think of them, right, like the sausage shape. That's what I call it, right? This is a little line, but, you know, I might draw it like a little sausage. A little fat guy like that. As it turns out, until chromosomes are replicated, you can't see them as a chromosome. They're referred to as chromatin. And that's what I call the spaghetti. Uh-oh, I can't spell spaghetti. Okay. So looking under a microscope during interphase, any part of interphase, all you'd see in a high power is spaghetti shape. You'd see no little sausages. Okay. The condensation or the coiling up of the chromosomes, that's the hallmark of prophase. So if you haven't gotten to prophase yet, you can't possibly see a chromosome. But we have to draw them so we can count them and talk about them and keep track of numbers. And so an unreplicated chromosome either looks like this little sausage guy or really lazy is just a stick. I mean, honestly, when I'm drawing them, it's just going to be a line. We know it's spaghetti. We know you can't really see it. But if I'm asking you, to give me a cell that has 5 chromosomes in it, please don't do this. I say, show me 5 chromosomes in interphase. Yes, they look like spaghetti in the real world, but I want you to give me, and it'll say somewhere in the text or in the question, please draw them as sticks. And somebody always draws spaghetti on the exam, and guess what? How many points you get for spaghetti? Yeah, 0. That's not good. Right? We all know it's spaghetti, but I want to know if you know what 5 chromosomes is. Seems silly, but we're going to follow the chromosomes through all these processes. And then the replicated guys, I'm just lazy and draws an X. Okay? And so on exams, it will tell you, please show unreplicated chromosomes as a stick or a line, show replicated chromosomes as an X. Okay, so that's very clear. Dr. Stein. Dr. Stein, I am going to have a written part on the exam. And it will be on paper, because we can't draw in Moodle. And it will be drawing mitosis and meiosis. So I'm going to tell you what it is ahead of time. And we're going to work through it in class. And then you're going to practice on your own, and you're all going to get all the points for it. Dr. Stein. Yes. It will be in addition to having multiple choice questions in a normal Moodle exam like Dr. Stein. You're welcome. All right, I'm just babbling. Let's move on. We're going to walk through. We're going to, yeah. Yes. Yes, I know. You typed in, right? Yeah. Okay. Yeah. But this is all about pictures. Like this whole section of the class is all about drawing stuff to show me that you understand it. Yeah, I'll try to remember to Moodle message, like pencil, I'll provide paper, I'll provide the question on paper, and then an extra scratch paper, so that, anything in the multiple choice that you need to scribble out, you'll have time for, that's all. Yeah. Yeah, and then you'll turn it all in, just like before. So and we'll talk, as we get closer, I'll keep, and when I'm drawing something and I say, ooh, this is very similar to something on an exam, what do you think that means? Yeah, because somebody is just staring at me. So I'm like, guys, you should be writing. So I'm writing. I'm not writing. Well, I mean, okay, I am writing because I like to write. But it's really because I hope you guys are writing it too. Okay, blah, blah, whatever. All right, here we go. All right, so the cell cycle. And now for pretty much the rest of this, we're going to talk mostly about mitosis. I'll throw a little meiosis in here off and on, but right, so if you're not sure which one I'm talking about, raise your hand. Just, I don't want to confuse anybody. I get all excited about chromosomes. Okay. So this is the cell cycle. And this part up here is really mitosis, okay, or the M of the cell cycle. It's a very short part of the entire cell cycle. And now remember, these would be cells like, we'll say in our bone marrow, right? We're constantly making more red blood cells. They only survive like 30 days or something, right? So you're constantly making a bigger supply. If you get any kind of bacterial or viral infection, you're going to pump out more white blood cells than your average. So it's constantly doing cell division, whether you're a baby, a kid, or an adult, okay? And so most of the time, the cell is actually in interphase. So the pink stuff, pink, red, brownish, whatever, all of that is interphase. So if the whole cell cycle took about 24 hours, which usually takes 48 or longer, but we're just going to make it up. We're just hypothesis, mitosis would be an hour of that. So 23 hours to do everything else, 1 hour to actually separate the chromosomes. Okay? So if you were going to look at a cell, somebody just drilled a hole in your femur, sucked out some of your bone marrow, slapped it on a slide and you're looking under the microscope, if we think about 1 hour and 24, that's about 1 out of 24 cells you actually see chromosomes. So the majority of cells on that slide are going to look like spaghetti, right, are going to be an interphase. Even if I tell you it's actively dividing, don't get sucked into thinking, ooh, all of them are going to have chromosomes because they'll be prophase, prometaphase, metaphase, anaphase, or right? The majority of them, you're still going to see an interphase. It's really difficult to find ones that are actually doing mitosis. Okay? And so that's one of the ways they look at a biopsy of a tumor to look for aggressiveness, how bad is it, is looking for its mitotic rate. Right? Compared to normal tissue, how many cells can you see the chromosomes? How many are in mitosis? Right? Cancer is just cells growing out of control, lost control of cell division, making lots of themselves. So if a normal liver cell takes 48 hours or more to go through cell division, a cancer cell may only take 12. And so then if you looked at that tumor under a microscope, lots more you'd be able to see in mitosis. So the mitotic index of the tumor, if you ever see a pathology report, God forbid, hope you don't ever have to look at one, that's one of the things. So the higher that number, the worse off, the more aggressive, the faster that those cells are dividing. Okay? And so this is supposed to be drawn to scale. Right? You can see interphase is huge, a little tiny bit of metaphase. Okay? And again, interphase is exactly the same for mitosis and meiosis. So we'll remind you of interphase when we do meiosis, whatever day that is, Monday. But we're going to do the meat of it here because it doesn't change at all. Okay? And so the three parts of interphase are G1, right, shown over here, S, and then G2. There's one other phase that I'm not sure if our new edition even talks about it, but there may be some weird test bank or weird Malone questions that refer to something called G0 or G0, G1. Yeah. So G0 is really what most of cells are doing because we're not actively going through cell division, right? Like we just take a big old punch biopsy out of your liver, right? Look at those cells. They're not even attempting to go through cell division. They're just doing their job, right? They're detoxifying all the alcohol you drank over the weekend, right? That's what they're doing. They're not getting the signal to divide. Using the term G1 to scientists suggests it's gotten the signal to go through interphase and mitosis, that it's in the process of cell division. Most cells are not in the process of cell division. They're G0. They're doing their job. The intro books try to make things simplified to not confuse you, which sometimes then confuses you later when suddenly a G0 appears in 360. And you're like, yep, Dan Malone didn't tell me about that. Right? So if we are talking about cell division, cells that are actively dividing, there is no G0 because those are the ones that are not dividing. So if your G0 or G1 is your genetic material chromatin or chromosomes? Chromatin, spaghetti, right? They haven't, when are they chromosomes? Yeah, once we get up here. So they're zomes with an M for mitosis. See how that works? And then everything else is chromatin, right? G1 or G0, chromatin, spaghetti. S phase has to be spaghetti because we're copying it. G2, still spaghetti. There's just twice as much content, DNA content, but it's still all spaghetti shaped. So interphase, G1S, G2, technically in interphase because they're going through cell division. If they're just hanging out doing their job, they're G0. Okay, and then we're supposed to teach you guys a little history, right? That's part of our job. And as it turns out, some of the GRE questions, biology GRE, talk about scientist names and what they do. I figure if people are studying for the GRE, that's something they should study, because if you take the GRE 4 years from now, you're not going to remember these people's names, even though they are important. But right, we need to know mechanism, I think. So anyways, this Fleming dude was one of the first ones to see the chromosomes under the microscope. And in plant cells, they're a lot easier to see than in animal cells. So a lot of the pictures that we see of chromosomes under a microscope are from plant cells. Mitosis means threads. That's when they can actually see the DNA material, right? Because if you look in G1, S, G2, G0, it's just spaghetti. You don't see anything in there, right? You can see the nucleus, but you can't see anything, right? No sausages. Okay. So I guess we're going to talk about S phase first. And it is one of the most important phases of mitosis because it sets up having two of each type of chromosome, right? Replicating the chromatin, making an exact copy of every single molecule of DNA. And in a chromosome, the DNA molecule is linear, right? It's this big, huge, long string of nucleotides, right? You guys did nucleotides in chapter 3 or something. And for DNA, it's a double-stranded nucleic acid, right? Nucleotides, one strand, complementary nucleotides on the other strand, right? Double-stranded. The two are anti-parallel. Did Stein talk about being anti-parallel? It's one of my favorite things ever, anti-parallel, which means one strand is the 5' end to the 3', the other strand has to be 5' to 3' going the other way, right? Because complementary base pairing happens when one nucleotide's right side up and the other one's upside down, right? A's and T's, G's and C's, one side's 5, one side's 3, that means this one's 5. I can't move that one, that one's 3. Right? The polarity, every single nucleotide has polarity, right? The phosphate end is 5', the 3' OH, and then we label the strand as 5' because if that nucleotide has a free phosphate hanging off, nobody's attached to it, this is the farthest side, that's the 5' end. Each one 5', 3, 5, 3, 5, 3, 5, 3, blah, 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 blah. The last one is attached at the 5' phosphate to the other 3' OH. But its 3' OH is dangling off into space, so that's the 3' end. Which means the guy complementary to it has to have his 5' sticking off, dangling into space. 5, 3 this way. Which means on this end, its 3' OH is dangling off. Okay, so that's the orientation of the double strand. Always anti-parallel, always complementary base pairs. And each chromosome is one long linear strand, billions of base pairs. Right? So for us, in a normal cell in G1 or G0, we have 46 long linear molecules of DNA. And so we have to replicate every single strand, every single chromosome in every single strand. So it cannot be condensed down already, because then the machinery to do the copying can't get in there. So it has to be out in linear spaghetti in order for replication to happen. So again, I want you guys to think about answers rather than memorize. Because you can memorize spaghetti S phase, and then I give you a little twist on an exam. And I'm not asking you the exact question I asked you in class or the exact way you heard it, and all you have is memorized S phase spaghetti. But you don't really know what that means, or you're afraid to think about what that means, and then you can't find the answer because I didn't tell you the answer to the way I asked that question. But if you understand that the spaghetti is a long linear molecule and that it has to replicate itself, Well, it's impossible for it to be like this because replication won't work. I know how replication works. It can't work that way. Can't work this way, can't work that way. Has to work this way. Right? But memorizing alone is not going to get you there. Lots of my questions, you will not know the answer looking at it immediately. Okay? That throws lots of people into panic because we are trained memorizers. I was a trained memorizer in high school, beginning of college. Oh, I could memorize the crap out of anything. Okay? You guys do need to know all the content, so I'm not saying memorizing is bad. You do have to know all the terms and all the BS, right? If you don't know that, you can't possibly apply anything. But you also have to be able to think about it. Because memorizing alone will get you the vocabulary, but the questions, you're not going to be able to go, boink, that's the answer for all of them. You're going to have to think about it and then know that if you've studied enough, you have all the tools. I gave you everything you need to answer the question, but I've never stated it in quite that exact way. And so not to panic and think, oh my God, I didn't study enough, I don't know it, because I don't know. A lot of them you won't know the answer right away, and that's okay. You just have to trust yourself that you've studied enough, that you have the tools, you think through the question, you get the right answer. I'll give some, I can't even think of one off the top of my head, but I'll give you guys examples as we go so it's not like totally, holy crap, I don't know what the hell I'm doing. Because the quizzes are supposed to be pretty easy. They're supposed to be definitions, memorizing, stuff you absolutely have to know. If you don't know that, you're totally screwed for the application stuff. Right? And that's also to help you guys study as you go, because I'm sure Dr. Stein talked about studying every day, especially in the summer, and repetition is better for your head. Small, half hour, all over the place rather than 5 hours at a time. Mixing it up, different ways of learning stuff, watching, listening, writing, quizzing, all that kind of stuff. So you don't get bored and just stare at papers. Well, I went through my notes. Okay, that doesn't do anything. That's one thing. That takes you 2 minutes. It's a good thing to do, but that's not studying. Okay. Oh, sorry. Okay. So DNA replication happens during S phase. They call it synthesis. Synthesizing something means to make something, right? Make more of something. Okay. So if we have, I'm going to make it easy, 10 chromosomes, 10 strands, DNA molecules in G1, then how many, well, during S phase, we have ... it's happening. How many strands would we have in G2? Right? We have to double the total DNA molecules. So DNA molecules are doubled. The DNA content, the amount of actual genetic material doubles. The thing that just makes everybody crazy is that the number of chromosomes don't double. What the hell is wrong with you, woman? Let's just even make this easier because I'm lazy. Let's make this 4. Sorry. For 4 DNA molecules, we get 8 DNA molecules there, right? So if this is G1, right, S happens there, we know it's spaghetti, but I'm going to draw it like sticks so we can keep track of the counting, okay? So we have the 4 DNA molecules, we go through S, and now we have 8 molecules. But we don't call them 8 chromosomes because they are attached at the centromere. So when replication happens, the new strands don't just float apart from each other and be like, yay, I'm free. They're stuck together to their template. And so what we consider a chromosome is that entity, including the centromere thing that's stuck. Now, it still looks like spaghetti, right? In the real world, if I wanted an accurate drawing, it would be that. But if we're trying to count and keep track and all that, we're pretending like we're looking at the linear molecules as if they're all stretched out into the 4 miles or whatever they would be. And so, for numbers of chromosomes, doesn't change. Yeah. Yeah, so you could say it's in chromatin. Well, technically it's still chromatin. It just has a partner on it. It just has its replicated partner. From G1, right? I said it's chromatin until it's condensed. So it's still chromatin here, it's still chromatin here. If we're pretending to be able to count chromosomes, right, it's 4 and still 4. Because the two sisters, the copies, are still attached to each other. Does that make sense? So this really twists some people's heads, right? Yes, there's twice as much DNA, but we still call it one chromosome. And as we go on, I'll reiterate that, and I'll show you, and we'll keep talking about it to make sure it gets in. Yes.
But they're not chromosomes until they undergo.
We don't technically call them chromosomes until they're condensed down. So they're replicated chromatin. But if we were going to count how many molecules exist that would condense down in the next step, right, it would be 4 in G2. So even if I'm calling it like generically, draw me how many chromosomes in G1, right? We know it's really chromatin and it's really spaghetti, but draw me 4 lines. So that terminology is hard to deal with when we're actually trying to count numbers and keep track of the strands. Okay, so the only time I'll be picky about that terminology is if it's actually in a question about what's considered chromatin versus a chromosome, like a quiz question or something like that, right? If I'm writing the written question, it'll just say probably chromosomes, because, and then I'll make it clear, please draw unreplicated as a line, replicated as an X, so that it's as clear as it can be, not trying to be tricky, not trying to, I could write, there's no chromosomes because it's chromatin, and not answer the question, and, no. And then come to my office and argue with me and blah, blah, blah. Right, but we're trying to follow the molecules of DNA to get through mitosis and through meiosis so that we understand how you end up with 46 and 46, not just we memorized what's in metaphase and what's in anaphase and what comes at the end. Like, understand the whole process. So then even if you forget the memorized stuff, all you have to do is think through the process because you understand it. And then you don't need to have all that memorized stuff. Because memorized stuff for just memorizing disappears next semester. Stuff that you've learned and thought about may come back to haunt you 20 years from now. Now, I'll apologize for that. You may come up and start talking about the process of mitosis at like some party, somebody's wedding, and everybody runs from you and you're really embarrassed. You get all drunk, you talk about anaphase, nobody wants to hear it. Yeah.
So the chromatin, G2 still chromatin?
Yeah.
That will pretend to be there?
Yeah. So really, like, let's see. Look at that. I'm just going to squeeze that over there. So in G1, this guy, right, if I was going to try to, it's spaghetti. It's still a linear piece, but they're all wrapped up, right? They don't just sit in your cell all straight, right? And then in G2, it's, the big, where this one's all like this, and then there's another guy. But they're stuck together, the two strands. But they're not condensed down into the little sausages, right? And the book shows you sausages all the time in all of these because they're just, if you draw the spaghetti, it's impossible to make a figure, right, and follow the chromosomes if you're just drawing spaghetti crap. Right? But I just want you guys to realize that's how it is, because it can't be smushed down or you can't do replication. When we get to the process of replication molecularly, you'll totally get it. Yeah, it has to be all spread out linear to get all the machinery in there to do the complementary base pairing. Okay. Yes. So again, then this would be, here's 1, 2, 3, 4 chromosomes, right? And then this is 1, 2, 3, 4. Yes, sir.
Would it be replicated by S or would it be
That's a good question. So if I'm asking you these kind of number questions, I can't really ask you what is the number during S, because it would be somewhere between 4 molecules and 8. Right? So if there's a crazy quiz question just to see if you guys get it, one of your answers might be, during S phase, what's the DNA content? It would be like 1.5. If it's 1 and 2, at the beginning of S, it might be 1.2. But, I'm not going to, 25 minutes into S phase, I can't ask you those things. We don't even know that. But it's somewhere between 1 and 2. More than in G1, less than in G2. That tells me you understand that that's the process when copying happens. Because when we do replication, it's a process. It's not like we have another strand. And suddenly, 4 minutes into S phase, boom, we have twice as many. Right? It's this long process that gets through all of the chromosomes. The big ones take longer than the small ones. It's 200 base pairs a second or something like that. So if you have chromosome number 1 is huge, right? The Y is, right? Yeah.
Are these chromosomes like still double paired?
Yeah, so then that's a great question. So each one of these squiggly arms, if we zoom in, is still double-stranded DNA.
What about in G1? Are they still double?
Zoom in, because I can't draw that small. If we're looking at the actual molecules, my double helix abilities should be good by now, right? Each chromatin piece linear DNA molecule is a double helix. Right? Here's an A on this side, a T on that side, right? Base pairing in the middle. That's one piece of chromatin. One chromosome that's like out in its spaghetti form, not in its compact when we call it a chromosome. Right? So then the second one would be the same thing, double helix, right? 4 linear molecules of double-stranded DNA, which, we cheat because if I had to draw that, that's all I do this whole class, I'd just get lazy. We do that. Because a DNA molecule is double-stranded. Complementary base pairing, because you guys learned that in Chapter 3. And so I'm relying on that information to carry you from here. If you had issues then, go back now. Because replication will kill you if you don't understand base pairing, how those molecules are set up. I know Stein drew 5' and 3' ends, 3' OHs, all of that stuff. If that gave you a hard time, the rest of this is going to give you a hard time. So go back now and go back to that and make sure that's solid, and that you understand. When I say a molecule of DNA means double-stranded, because there's no such thing as a DNA molecule that's single-stranded. You can have single-stranded DNA transiently, but anytime it's in its molecule form, it's double-stranded. And so then this guy, which is why it makes it even harder. Here's a double strand. Here's a double strand, and they're attached at the centromere. That's the one X. Yeah, so during S phase is where, right, they transiently become single-stranded and we start replicating, getting the double strand. And then, right, we make a whole new piece here, a whole new piece there. It's attached. It looks like that. And we'll walk through replication, I think, after mitosis, meiosis, right, the actual how that works. But that's how you go from one double strand to two double strands connected together. Right, and so then there's 3 more over there, which will take me forever to draw. Does that make sense? That is bueno? Okay. So for now, we're just knowing S phases, replication, copy the DNA. As it's done, we have twice as much DNA content. If we're counting chromosomes, it's the same number of chromosomes. Right? So if in our cells, we never have more than 46 chromosomes. We have, in G1, we have 46 chromosomes. In G2, we have 46 replicated chromosomes. But we don't count them as 92 because they're stuck together as an entity. Right? So we never get more than 46 chromosomes. Yes, we have 92 molecules, right? 92 double-stranded linear molecules, but we count them together because they're attached. They don't live apart as a chromosome. They're codependent at that point. They need therapy, which they're going to get during mitosis. They're going to be able to break apart and live on their own. Okay, so then G2, they're named as the gaps. Originally, nobody really knew what the hell was going on in those. Now we know that the cells are getting ready in G1 to go through mitosis, and G2 is mostly making sure we have all the organelles replicated and enough stuff going on, a big enough size cell in order to go through mitosis. So if we think about, here's a cell, right, we're going to pretend like we're in the liver again, because we love our liver. It's good for us. Except it's not really, it's somebody else's liver that only has 4 chromosomes in it. I'm not drawing 46. Okay. And we're in G1. Okay, this is the normal size of a liver cell. If the cell didn't get bigger, then after mitosis, right, S, G2, M, cytokinesis, each cell would be half the size. And then it would have to grow to get to be normal size. And then if we need... So actually what happens, instead of that, here, let's go like this. So we don't get confused later and go, what the hell was that woman talking about? Yapping on and on, I was sleeping, I lost her. All right. This guy sits here, and then whoa. He gets bigger, so that after all the other BS, sorry, it's supposed to be the same size as that. They're about the same size as the original, yeah. So G1 and G2, growth. G1, a lot of the important stuff is, do we have enough energy, machinery to get through synthesis? Right, we need ATP, we need all the nucleotides, right? We also need, if we think about, you guys did organelles, was she super excited about organelles or what, Dr. Stein? Did she do vesicles and just like get frothy? Yeah. So if you think about every time a cell splits, you can't half the organelles. Okay, now you only have half the amount of ER. You only have half the amount of this, half the amount of that, right? We want to set forth these daughter cells, the offspring, ready to thrive, not at a disadvantage where they're smaller with less stuff, then they have to try to survive and get bigger and do their job, right? We want the cell to do all of that before it replicates, because if the cell can't do that, it stalls cell division. That means there's something wrong with that cell. It's not a healthy, hearty cell. That's not one we want more of. We want the ones that can make more of their organelles, grow in size, be all healthy, right? go through the cell division, mitosis, cytokinesis, have two full-size, fully functional cells ready to do their job. Not two little wimpy, I got half of my crap, I got all my DNA, but that's it. And now I have to spend all this time building everything up before I can detox your blood, right? By then, you're just drunk forever, and you're just laying there in a coma, right? You want your cells ready to go as soon as they go. So G1 and G2, G1 getting ready for replication, G2 checking to make sure everything's fine, making sure we have all the organelles replicated, everything ready to go before we go to mitosis. Yeah? No, this is still part of G1. So this is a so you can think of, we start this size, we're getting bigger, we're getting bigger, we're getting bigger, right? We go through mitosis, we can still get bigger at that point in G2 as well, and then so that we end up with both of the same size at the end. So it's really just, growing, replicating all the crap, being totally ready so that once we replicate the DNA, go through mitosis, split in cytokinesis, two regular-sized cells, the same number of organelles as the parent also. So when they say an exact genetic copy, yes, but also an exact copy of at least almost the same mitochondria, almost the same, ER, Golgi, all the stuff the cell needs. Yes, sir.
So G1 right there is the one that has replication already in it?
No. So G1 is before replication happens.
And the split right there, would that be the replication part? Or
Yeah, so in this one, just like here, so S would happen here, G2 had to happen, mitosis had to happen, and cytokinesis had to happen in order to get to that end point. Right? This little picture was just, right, this guy is supposed to be the same size as these two guys. Right? If this one had 12, if the first one had 12 mitochondria, each one of these guys has 12 mitochondria. Not each now has 6, and they have to replicate once you get there. It's supposed to all happen up front. And as far as we know, for the most part, it all happens up front. So the gap phases, there's lots of stuff going on. Again, 800 years ago, when I took this class, they were like, it's just resting. Nothing's happening. All the actions in in S phase and mitosis, just ignore those little, we don't know. But now we know what's going on. Okay. Right. So that's the gap, right? Organelles, more cytoplasm, get bigger, right? G1, which is really technically G0 when they're doing that, but I won't be picky about that. But in case you read that in the future, you'll know what that whole baloney was. Okay. So bacterial cell division is not mitosis. Why? What do we know about prokaryotes? Prokaryotes. No nucleus, right? What does their chromosome look like? It's a circle, right? So mitosis refers to linear chromosomes. So we never call anything that has a circular chromosome going through mitosis because it doesn't. There's no phases of circular chromosome coming apart, right? Because there's only one of them. And so again, see how they're showing this as spaghetti? If I were going to draw it so we could actually see what it looks like, right, I'd say, here's my, I'll just draw all these first. Okay, so there's your chromosome, right? And again, like your question, it's a molecule of DNA. So it's double-stranded, right? So we draw the line as the molecule of DNA, but we know molecularly it's a double-stranded circular molecule. So I can't draw the, And in simple cartoons, the books don't always draw the, but DNA is always double-stranded, whether it's eukaryotic or prokaryotic, right? So by definition, it has to be a double helix. Anti-parallel, 5' and 3', except there's no ends. I can't make a circle, right? There's no ends to it, so there's no 5' end, 3' hang, because it's a circular molecule. So when they say replication happens, to draw, oops. Here's a double helix. We do replication, right? Again, like somebody asked, yes, they transiently come apart, and you get in there and do complementary base pairing all around, right? And you end up with now 2 double helical circular molecules of DNA. So that would be analogous to G1, S, right, end in G2. But we don't use those terms in bacterial cells because they don't go through the same processes. Why don't they replicate organelles like eukaryotes? Because they don't have any. Right? There's a trick question. That would be mean. Right? Why do they not need to replicate organelles? Why don't they need G1 and G2? Because they don't have anything to do. They need to grow a little bit, right? But in some cases, bacteria actually do get smaller when they go through binary fission. They don't always grow bigger than split in half. It depends on the environment. Okay? And then just those two, right? Here's this double helix and this double helix. They move to the poles, and then it just pinches off. Right? These are still double helix, double helix, double helix, double helix. Right? And now we started with 1, and now we have 2. And this is called binary fission. Binary meaning 2, and fission meaning splitting. Right? Fusion is coming together, fission is pulling apart. So it's not mitosis. It's binary fission. Prokaryotic cell division we call binary fission. Eukaryotic cell division of somatic cells we call mitosis, interphase mitosis, cytokinesis. So are the daughter cells identical to the parent? Yeah, barring any random mutations that might happen, right? Random mistakes in replication. Sometimes replication makes some mistakes, right? But barring any of those, it's identical, right? Which also means that it's a clone. So they don't do sexual reproduction, they do asexual reproduction. And so all of the offspring are clones of the parent. That is pretty much all we're going to say about prokaryotes. Because really, that's all they do. But they do it really well, as it turns out. Oh, I'm probably good by now. I think I was charged. Okay. So if we look back at this, this is what we call a karyotype, which is looking at your chromosome. So a chromosome spread. In this case, we're looking at normal human karyotypes. Okay? And we look at these guys. Do these guys look like spaghetti or do they look like sausages? Sausages. So if you were going to take a gander at where they are in the cell cycle, where would you guess? Yeah, prophase, someplace in mitosis. As it turns out, it's easiest to pull the chromosomes and get a good picture of them right about metaphase. Right? So a metaphase spread is usually what they do for a karyotype. Okay, so just, you don't need to know that. Fun fact. Okay. And then in this case, these are colorized, right, so that you can see the different chromosomes. If they're doing a stain, the chromosome 1 has a particular pattern of staining if you use a specific kind of dye. If you're using fluorescent probes, like they can actually label specific genes. Right, so if you throw this dye onto a whole bunch of jumbled up chromosomes, spread them out as best you can identify what the 2 number 1s, the 2 number 2s, 2 number 3s, and then they're photographing them and then photoshopping them together. So they're not actually like taking molecular tweezers and moving chromosomes next to each other to spread them out like this and take a picture, right? They're doing that on the computer. Okay? They used to actually take a photograph and then cut the pieces of the picture and glue it. That's real cutting and pasting. Yeah, that was like too many years ago. Okay? And so you can see that the banding patterns, the color patterns are identical on the pairs, right? And would we say, does it tell you now, is this a boy or is this a girl? This is a boy. How do we know? Look at that guy. He's so cute right there. Right. If it were female, there'd be two X's. Right? Boys have an X and a Y. But we see there, oh my God, I can't see. Two number 22s, two 21s, right? All these guys. Two of each. Two sets of chromosomes. Two number 1s, two number 2s, two number 3s, all the way through an X and a Y. The genes on chromosome 1, on this one, are the exact same genes on that chromosome 1. The gene for earlobe shape. How your earlobe is shaped. I don't know which chromosome it's on. I'm making it up. I'm pretending it's on chromosome 1. It's a big guy. It's got thousands of genes. It could be on that one. Okay? You have two copies of every single gene. Unless you're a boy, and then you guys are missing the second copy of the X gene. Okay? You're a little low on your DNA, sorry. Okay. That's another thing that everybody nods, yes, I get it, and I talk about it, and I talk about it, and then I ask it on an exam question a little bit differently than the way I talked about it in class, and we don't know the answer to that, right? Because, and that's just common, that's just how we are raised, that's just the media, that's just what people think. We talk about the gene for whatever, not the genes, right? You have the, gene for whatever, and your hair color gene. We're never talking about them as pairs, as lay people. We only start talking about each trait and each protein in your body. Actually, there's two copies of each gene here, and so that throws people. But there has to be, because you have two number 1s, and number 1 has the exact same genes on it as the other number 1 you're carrying, right? Earlobe shape, whether you have a straight forehead, hitchhiker's thumb, tongue rolling. What kind of collagen, all the different things, part of one of the eye color genes, one of the hair color genes, right? The exact same gene. Two copies of every single one, because chromosome 1, one of chromosome 1, who'd you get from? Mom in the egg. The other chromosome 1, then you had to get from dad in the sperm. So you've got the gene for earlobe shape from dad and the gene from earlobe shape from mom. What you look like, your phenotype, depends on how those two genes interact. Okay? And then we call them alleles, that stupid word allele that just makes people's underwear get all in a twist. Okay? So I'm just going to bring it up now because it's going to come back to haunt us in the genetics portion, and the more we talk about it, the more comfortable we get. Right? Your alleles do not have to be exactly the same. It's the same gene, the gene for earlobe shape. Mom gave you the allele that's all flappy. Oh, right, the big fat guy laying down here. Oh. Maybe dad's gene for earlobe shape is his earlobes are tight against his jaw, right? They don't curve in and make this big fat blubbery thing. They're just nice and fancy tight. Okay? How those two then interact determine whether you have floppy or tight. But it's the same gene for earlobe shape. It determines when you're in utero what happens. Right? It can, which is bizarre. There's a gene that controls that. Like, really, who would care? But apparently sometime in evolution, right, it must have been sexually selected for or something, because everybody's either got flappy or straight, and a little bit of in between. So as it turns out, the big flappy blubbery guy is dominant. So if you had one of each, you're going to look flappy. The only time you have the tight against your jaw is if you've got the tight allele attached from mom and dad. And then your genes would be exactly the same, earlobe shape. Your alleles are the same, attached, non-blubbery. Okay? So every single gene we have two copies of. One came from mom, one came from dad. You might not see the phenotype from, like, my nose is exactly like my mother's. It has nothing to do with my dad. I still have nose-shaped gene from dad. It just lost, right? Lost out during development, did nothing. My nose is the exact same shape as my mother's, which is the exact same as my grandmother's. And it's not good, but that's the way it goes. Okay? My daughter left out. She did not get my nose. But that doesn't mean I don't have the gene for nose shape from my dad. It's just the allele lost out. The mom allele was dominant. I got the mom nose. E-slope just... Okay. And so because we have two sets, or two copies of every chromosome, two copies of every gene, therefore, we call ourselves diploid. Ploid is your sets of chromosomes. So ploid just means set. Or your ploidy is how many sets do you have. So our ploidy would be 2. Di, right, meaning 2. Ploid, meaning set. Diploid is also known as 2N. N as the ploid part. Set. 2 sets. The N stands for the set. Okay. If there are some plants out there that get all crazy and they have more than 2 sets of chromosomes, animals can't tolerate more than 2 sets of chromosomes, right? That just doesn't work. But plants can. So if they have 4 sets of chromosomes, then they'd have, oh, that's probably not going to show up, another, and each one of these is really replicated with its centromere. They'd have 4 of each. And then we call that 4N, or tetraploid. Octaploid would be 8. Strawberries are octaploid. Okay, so that's your ploidy. So we're going to be talking about mostly diploid stuff because that's what vertebrates and most animals that we think about are. And plants, lots of plants are diploid. There's just some that can tolerate having more chromosomes. Most of them are the agricultural ones that we've selected for having more sets of chromosomes because it results in big, juicy fruits. Right? The wild strawberry is like this teeny little tasteless red thing, but like go to, get the strawberries from the farmer's market and there's these big, that's because they have 8 sets of chromosomes. Pumping out the goodness. Okay. So this is just the same stuff that I said, right? The N, again, meaning the sets. And so if you only have 1 set, you are N, and they call that haploid. It should be uniploid. That would have been much easier, but some ding-dong, or monoploid, right? But so some a-hole named it haploid just to mess with us way back when, and it stuck. I like uniploid better, much easier for students. Okay. So there are organisms out there that exist as haploid organisms, just not the normal animals. Weird, some insects, some weirdo single-celled protist type things can exist as haploid. What are there any cells that we have that are haploid? Yes, the sex cells, our gametes are haploid. That's the whole point, right? We go 2N to N. And then when you put 2 N's together, woohoo, you go back to 2N. Right? That's our whole point. So we have to make our gametes haploid in order for when fusion of them happens, we go back to diploid.